from Microbe TV, this is TWiEVO, This Week in Evolution, episode number 72, recorded on October 20th, 2021. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey Vincent, great to be back with you on the airwaves. We're up and running again, live on YouTube. Greetings, everyone.
Yeah, hi everybody. Got some hardcore scientists here who were at my virology course this morning, and they're now here, and some of them are probably going to be on Q&A with Amy tonight.
Wow, that's a triple header. That's a rare breed of science enthusiasts there. Hey, Vincent, I'm looking at your background. It looks like you have some new setup running.
Oh, so I'm in the incubator, yeah. And I've been recording here for the last 2 weeks, and it's been going well. There's some glitches, but we'll get through them. And I like it. It's pretty cool. The time goes by quickly. And I came in this morning, I had to assemble a wire shelf to put all the stuff that I have accumulating in here. And then I had to vacuum the rug because it was a mess. We're opening all these boxes makes a mess. But yeah, I like it. The background could change. As I showed on TWiEVO yesterday, I have this watercolor that Nels is echoing, they're saying. Nels, you got some... maybe you should use your headphones, dude.
All right, I'm going to go to the headphones here. I don't have a slick setup like Vincent does.
Maybe I need to come out there and do some consulting and set you up properly, you know.
Yeah, that's right. I could use a little upgrade. So I'm actually, let's see, I think I need to switch this. Hopefully this will help. I'm in the middle of an upgrade myself, so I'm coming back from temporary LD Lab studios here. We're doing a little bit of an upgrade in my old office, so it's probably the perfect time to also some improvements on the audio. Is that helping? Do you think the echo is gone?
So chat, tell us what's going on. Let's see what they say in the chat. The echo is kind of fun.
Sort of an echo chamber, an echo of evolution.
Reminds me of... you don't have your laptop playing the chat, right?
It shouldn't be. So I was trying. So I've been on our live broadcast. I've been flying blind a little bit here, meaning that I'm not following on YouTube. I'm kind of excited though to sort of see the conversation as it unfolds. So
yeah, you still got an echo. So
still have an echo. Wow. So I closed that window.
Now you're hearing through the headphones, right?
Correct. I hear you through the headphones.
You're not. It's not coming out of your computer, huh?
Shouldn't be at this stage, unless I somehow still secretly have a window open here. This is the joys of live broadcasting, Vincent.
Yeah, some people are saying maybe you have two windows open, so check that out. What are you doing this on a laptop?
So we're on a laptop. I've got just through Chrome is our single connection.
This is through this software that I'm streaming with, right?
Yep.
And you have nothing else open.
I'm going to close. I'm going to quit. Actually, let me try this. So I just quit Safari.
Good. Nothing else open.
So it's only Chrome, one window.
No tabs. No other tabs in Chrome open.
No other tabs. One single tab.
What say ye, oh loyal chatters?
Are we still echoing across the divide?
Yeah, I guess we have to talk in order for them to
That's right.
to hear an echo or not.
Yeah.
Is Nels still echoing? So Nels, how's the weather out there?
Vincent, we've just been getting the first signs of snow as we transition into the fall to winter calendar. Still echoing?
I don't have an update. Yeah, it's still echoing.
Wow.
You want to leave now?
Tell me to hang up on you.
Hang up and come back. Yeah.
Yep.
And then if that doesn't work, we'll replug the mic. Isn't this is reminiscent of Q&A in the early days when Amy had echo or I had echo, actually. It was me. It was my fault. So Nels is maybe, yeah. He's restarting the streaming program. And then we'll have him, he's got his USB mic. Maybe he's actually not using the mic. He's using the laptop mic. We'll sort that out. As I said this morning, we solve problems. We're scientists, right? Okay, here he is. And we will put him on as guest number 1. Let's see. I have no sound from you at all.
Moment of truth.
Oh, okay. Now, Nels, tap your mic. I want to hear what it sounds like. Okay, so your mic is actually working.
That's good
news. And the echo is Vincent's mic picking up Nels's audio. No, it's not, because I have earbuds in. There's my.
Oh wow.
Nels's sound is not going into my.
You have invisible gear there.
My microphone. Yeah, I have these very small earbuds that is not. There's no way that that's happening. Is your laptop mic on? So in the software, go to your system software. And make sure that you've selected the headphones and the external microphone.
Yep. I'm going now to sound. It is on the mic. Output is microphone, and input is. Here we go. I think we might have solved it.
Because you're using the ATR where the headset's plugged into the mic, right?
That's correct.
So should be both in and out. The mic should be in and out.
Yep.
Okay, folks, what do you hear from Nels? Nels, you having good weather out there? It's snow. Did it snow yet?
It has. There's snow on the mountains, as the famous saying goes.
You think the headset has a mic in it? It's just a headset. Those are just headphones, right? There's no mic in it.
Just standard headphones.
Have you always used those?
Yep. And we're on ATR for both input-output.
Mm-hmm. Mm-hmm. I don't know what to say then. How about if you the mic is a USB mic, right?
That's right.
Can you unplug it and plug it back in?
Yeah, I'm going to cut it right now.
So I'm not hearing him when he turns off his USB mic, so it's not coming through his computer. Yeah, so that cut you off.
No, I'm back on.
And what if you... It must be just that you're in Salt Lake City. That's where the
We found a wrinkle, an internet wrinkle. We've done this plenty of times. I would say 4 or 5 times without a eCam echo.
Yeah, it's weird. I don't believe it's my end. Let me just check my things here. Hold on. So my input is my thingamabobber. Let's check it out. I'm sorry to be. Output is F series. Input is F series. No, it's.
This is important to clear up.
All is good on my end.
Huh.
It's still echoing apparently.
Wow.
So let's try something else.
I'm tempted to do the classic just total reboot on my computer. You game for that?
So let me try this. They're thinking it's you coming through my headphones, but I'm wearing earbuds. The ear sound isn't coming out of here. But let's just do an experiment. Yeah. I am going to mute my microphone and you talk. Okay?
I will.
So I'm going to mute in 3, 2, 1.
All right. One of the rare times when Vincent is speechless because his mic is cut off. Still echoing? I'm still talking. So big apologies to everyone. I think we have 58 of you who are patiently trying to help us troubleshoot our audio echo situation, which continues to persist despite a few measures here to try to change our settings. Still can't hear you, Vincent. There you are.
They're saying it's perfect when I'm muted. So you just do the show now by yourself.
We're going to have to do like a When I'm talking, I'll turn my mic on. When you're talking, you turn your mic on situation. Is that where we are at the moment?
So no echo when Vincent is muted. How about now when I'm not muted? Go ahead, Nels, talk.
Yep, I'm still here helping to troubleshoot. Am I echoing, everyone?
How about if you lower your mic volume, Nels?
Oh, yeah, I can do that. Lower the mic volume.
I don't know how it could be me, folks. I have earbuds. There's no sound coming out of this room.
Yeah.
You know? Unless it's escaping unbeknownst to me. Go ahead and talk, Nels.
Okay, I am back to my input, which is input volume is coming down. I just turned it half.
All right.
And now it's down to a quarter. Back at half volume for the input. It's kicking up automatically to try to pick up on the microphone.
I can't move my mic too far away, otherwise you guys are not going to hear me. But I find it hard to believe that Nels is coming out of my earbuds, which are really tight in my ear, folks. They're all the way in the auditory canal, you know? The auditory canal. Vincent will have to mute and unmute himself for now. So let me ask you for one last time. Nels, what's the weather there?
One last time, we've got snow on the mountains here, Vincent. Snow on the mountains. That's what I'm reporting.
I can lower my headphones. This weekend echoing. Let's try that. Go ahead, Nels.
Okay, still here. Excited for our episode once we can get our audio situation figured out here and hopefully control this unexplained echo.
That's the lowest I can go and still hear Nels. I don't have a mixer. I just have an audio interface like Nels. Nels has a mix pre. I have a Zoom F6. So there's no mixing going on.
Interesting.
Let me try one more thing, folks. Nels.
Yeah.
Let me try one more thing. Finder.
Absolutely. I'm going to keep talking while you're adjusting the controls.
Why don't you start the first paper?
Yeah, great idea. So I'm going to skip through, just given in the interest of time here, we had a quick SARS-2 evolution update. We're going to, I think we'll sidestep that. Nothing too major, just kind of keeping an eye on the rise of the Delta variant and some interesting patterns. If you go to nextstrain.org and just even look at some of the high-level sampling going on there, it's really kind of tells a story of evolution in action. So big recommend to take a look there. Our game plan today is to do a deep dive into the TWiEVO mailbag, actually. A couple of short letters that I think are really good suggestions for us. And so that's sort of on the menu today. And so I'll just get us going. So Mark writes, "Dear TWiEVO team, I would be keen to hear your explanation of this journal article. All the best from just a bioengineer." And this is Mark. So this is our first paper. It's the title of the paper, and we'll have the link to this, is "An Ancient Viral Epidemic Involving Host Coronavirus Interacting Genes More Than 20,000 Years Ago in East Asia." And so this is recently published in Current Biology. An interesting paper. So this will connect us to some of the coronavirus evolution, looking at it from a slightly different angle here, which is, are there clues even in sort of human history that might teach us about, past epidemics that could have involved viruses similar to coronaviruses and even SARS-2. And so the authors here, it's a team from several different international teams. So some scientists from Australia, in addition to several other locations. So we've got the headquartered at the University of Arizona. So David Enard is the senior author. Yassine Suleimai is the first author, with some collaborators from both Australia and then also California as well. And so the reason I think, and so I'm glad Mark picked this up. So the reason I think this is interesting is because, this takes a slightly bigger view of coronavirus evolution. So that nextstrain.org view that we've been spending, you can kind of gaze from day to day and watch the pandemic unfold in real time. Is there a way to sort of tackle this from a deeper perspective? And so that will be the first half of the show, is discussing this. And then we'll actually do a dive on the second half, into even deeper evolution. And following a number of requests, including from the mailbag, we're actually going to step away from coronavirus evolution and think about sort of evolution of early cellular life. Okay, so the broad question here: do genes encoding proteins that are involved in SARS-2 replication show signs of selection? Is there an evolutionary signal that we can deduce from this? And so the authors have some previous work, because it's coming out of the Enard lab, just south of the border here from Utah down in Arizona, and where they've basically launched this idea that you can categorize a number of proteins encoded in our genomes as VIPs, or virus interacting proteins. And so what they do here is they actually take some other work done at UCSF in San Francisco. This is Nevin Krogan's lab, which is to do some in vitro experiments where you try to connect the interacting proteins, so host proteins that directly interact with proteins encoded by SARS-2. And so this leads to a collection of maybe 50 or 100 VIPs or VIPs. And those proteins are then, or the genes encoding them are then used to look for signatures of selective sweeps. And so this is different. So already actually from that original mass spec paper that was published some time ago, there was some evolutionary analysis sort of folded into that, identifying key factors that might be interacting with virus proteins, and then asking, if we take a big view of evolution over, say, millions of years, are there signs that those host proteins have repeated signatures of adaptive evolution of so-called positive selection? We've talked about this a lot on TWiEVO in the past. So this is this DN-DS ratio, where you're comparing the rates of the appearance and retention of non-synonymous mutations, where you change amino acids in the protein, compared to silent mutations, where a mutation still encodes for the same, or is the same codon for a different codon for the same amino acid, I should say, a silent mutation. And so the answer is yes, kind of from that view of evolution. And then this paper tackles a more shallow sort of swath of time, and actually asks, in modern human populations, do you see signs of selective sweeps? And so there are two tools here, and then we're going to get into some alphabet soup. We won't spend too much time going into the weeds, but so two population genetic tests where you're looking for signs of selection, and we'll talk more about that in a moment. But so the first one is the so-called Ihs test, or integrated haplotype score. And this sort of arose maybe 15 years ago, something like this, just as we had completed the first human genome. And then what was the next thing we did? We started sequencing a lot of human genomes and comparing them. So early days of the so-called HapMap project, and Hap here is short for haplotype. So basically beginning to compare not just sort of single mutations as you sort of march along a chromosome, but collections of mutations, and seeing where they match up between different individuals' genomes. And so the IHS, or the integrated haplotype score, took advantage of the fact that actually there's some information that you can glean from just comparing these stretches of chromosomes by comparing them between humans, and asking, how are the patterns changing in terms of the shared mutations? And so the idea here, it sort of ties back into this, again, into the selective forces that are going forward here. So natural selection acting on selecting for a mutation that's beneficial. And so in the context, or in the landscape of the chromosome, what will happen is even a single change that might be beneficial, you might enjoy some more protection against a virus infection, or something like this, will stick around. And coming with that is a number of of other mutations that might have just appeared roughly at the same time by random chance, and have sort of this unique pattern then of mutations. And these other mutations that don't provide necessarily a benefit, but are so-called hitchhikers. They hitchhike along for the genetic ride here, so to speak. So this IHS test is a way of measuring or comparing these blocks of relatedness. And the idea is that given enough time, those haplotypes will get broken down. So as recombination is happening, those sort of shared or hitchhiker mutations will get pulled away or will recombine away from that beneficial mutation. And so this gives potentially two pieces of information, sort of as you find or propose a mutation that's under selection. The shorter the haplotype block, the longer the time has gone on. And so if you have a longer or a big haplotype, this suggests a really recent sweep, where in the population level, this is sort of a new combination of now shared haplotypes that have shown up in the recent past. So that's one of the tests. The other tests, I won't go in kind of as deeply into the weeds. This is the NSL, number of segregating sites by length, which does roughly the same thing, actually. It's slightly more sophisticated. So you have to take into account that mutations in real life aren't totally random. There are sort of recombination hotspots among chromosomes. And so you're trying to correct for those kind of background signals that could otherwise, sort of cloud the interpretation of selective sweeps by both of these tools. Okay, so basically, what the authors decided to do was to just take this set, for the first time, take this set of protein or genes encoding proteins that were proposed to be involved interacting, involved in SARS-2 replication. The virus proteins are interacting with these host proteins, and then run these tests and in different populations, human populations. And what they see, or what they propose, is an enrichment of selective sweeps in East Asian populations relative to any other human population. And I would put a couple of caveats here that I think are important to consider. So one is actually just this definition of VIPs. For a couple of virologists like Vincent and me, it's sort of tempting to think that viruses are sort of running the show here, and that all proteins are are VIPs. Our proteins are here for viruses to use to take advantage of in the evolution of our in their evolution to propagate themselves. Of course, no gene from a host is sort of dedicated, or you hope, isn't dedicated to just propagating viruses. And so all of these genes, and we might return to this in a few minutes, have day jobs. They have other functions that might benefit host reproduction ultimately, even the very sort of mundane jobs of day-to-day cellular activities, moving around membranes, dividing cells, building different structures, kind of conducting cell biology. So I think that's worth keeping in mind, is that, no protein is truly a VIP. Proteins are proteins, and then some might get entangled in some cases with viruses. But I think it's worth it to remember not just sort of the specific proposed interaction, even in this case with a very specific virus, but that proteins evolve under many different, selective forces inside and outside of virus infection at all. And so a little bit of a caveat there. And then a second one is that as we think about these virus interacting proteins, and look at the selection, we're not just talking about single ones where we're looking for selective sweeps. We're asking for an enrichment among the whole class relative to all of the other proteins in the genome, or similar sets of randomly described. And so if you look at the figures that are proposing this enrichment, actually the confidence intervals are pretty massive. And so is there really an enrichment at all? Could be an open question, even from sort of a statistical standpoint. But that said, so there certainly was something different about the patterns of sweeps or enrichment of sweeps among these proteins in East Asian populations. And then the authors did something clever here. I think they went to another tool that sort of asks the question about selective sweeps in a different, in sort of a different framework. And that is, when are these things happening over time? And so there's a tool called Relate, which is newly published just from a couple of years ago. And this, in short, is a pipeline in order to take these comparisons, these haplotype comparisons, so these are the genomes from sampled human populations, and then try to estimate the date when that selective sweep happened. And so this uses something called ancestral recombination graphs. So you're trying to work backwards in the sort of generation to generation in order to infer when in time the haplotype switched. And you can kind of do this because you have, as you build any comparison of phylogenetic. Oh, fantastic.
I think I'm back. Do you hear me?
I can hear you. loud and clear.
All right.
Now the question is, are we echoing?
They say I turned off my mute. You're not echoing. So I did a little. See, I did a in-flight adjustment.
That's right. This is real-time live podcasting, science show with no safety net.
I'm not typing, Steph.
So, Vincent, can I ask what was the glitch here that had me echoing?
So, the way this setup works is my microphone goes into an interface, right?
Yeah.
And the interface is plugged into the USB port on the computer, right? I have your sound going. This interface has 6 channels, so I have your sound from the USB going in, so I can hear it in my headphones. But apparently that's no good, and so what I did was turn off your input on the interface, and I plugged the headphones into the computer. So there's some issue in the interface. As someone pointed out, it's not really a mixer, but it's sort of.
Gotcha.
But it works, and that's very puzzling. However, this paper is cool. Go ahead, Nels. I'm sorry to.
Yes. Not at all. So Vincent, let me catch you up. So we're kind of in the depths of Figure 2 here.
Okay.
Where we've already proposed that this collection of proteins, genes encoding these proteins that interact
VIPs
with SARS-2, the VIPs,
right,
could have some interesting signatures, especially in East Asian populations of selective sweeps. And now the authors are proposing a date for this. So they run these recombination graphs where they're basically working backwards in haplotypes as you're comparing them and seeing how recombination is breaking up the patterns in different ways.
Right.
To actually, say, and they identify a window, a 200 generation window.
Right.
And let's have a little context here. We think in terms of like 2 and 3 generations to our grandparents, maybe, 4 generations to our great grandparents. So now, step that back in evolutionary time. And so this dates roughly the 200 generation window where they see an interesting signal for these sweeps, these haplotype patterns. Again, in East Asian populations is about 800 to 1,000 generations ago. And so
all right, so the people want to see. I'm going to put up Figure 2 here. Let's see.
Good idea.
I think that's That's number. I put the wrong input in. All right, so there's Figure 2. Let me blow it up a little so you can see it, and I'll center it. There's Figure 2, right?
Yeah, fantastic. So, and what we're looking at here on the x-axis, moving from left to right, is 0. That's today, this generation,
right here,
and then stepping to the right exactly, back 1,000 generations, 2,000 generations, 3,000 generations, and there's 2 graphs here. So we're looking at a density, and again, this is another maybe caveat here is we're not measuring any sort of single protein, or we're not trying to infer any sweep of any single protein, but sort of an enrichment of now these in kind of pinkish color, reddish color. These are the VIPs. I think it's just like 40 or so virus interacting proteins, and then this is compared to other genes in purple.
Is that the whole proteome? Everything else, Nels?
Yeah, good question. I can't remember if this is actually scaling to roughly the same number of genes or the rest of the genes. Oh no, it's all genes. Yeah, so that's the rest of the proteome. They're very different, similar words with very different meanings. And so what you can see is that there's no enrichment of the density, which is on the y-axis, is sort of the number of genes that are showing these sweeps. And so this is relatively quiet. And then there's this one blip among the coronavirus VIPs that sticks out from 800 to 1,000 generations ago. And this is, or 770 to 970, centered at 870. And this is, I think, pretty interesting. So what it implies, and what the authors are, kind of speculate on, is that there was a pretty massive pandemic, probably, given a lack of any sort of, medical interventions, and this is in our own species, our ancestors, Homo sapiens, a pretty massive, maybe an echo of a massive selection event where basically many genes in this category were sweeping over the course of these couple hundred generations. And so, that's kind of part of the, almost philosophy of these host pathogen interactions, but to actually, kind of try to identify a specific event, like a catastrophic event in human history like this, to infer that from these signals. This is, I think, probably the most novel part of this paper, of this study. And so, again, I want to put in a kind of a big grain of salt that VIPs aren't there just for the viruses. There's other selective signals. And so I think it's worth thinking about alternate hypotheses. And so I'm going to actually, jump ahead here kind of in the interest of time to first of all, let's take a quick pause at figure 5.
Okay.
And this one is relatively easy. So this is looking at perfect. So in both panel A and B, so the panels are two different East Asian populations, CDX and CHB, and I'm forgetting the
So
exact.
CDX is the Chinese Dai 1,000 genomes, and CHB is the Han Chinese from Beijing 1,000 genomes.
Yep, perfect.
How cool is it that we have these resources to probe, right?
Yeah, and that's another thing that I think this paper kind of like landed at the right time, where it took a number of tools that were sort of relatively recently developed in population genetics, and then put that together with sort of this growing wealth of new genome scale data, exactly like you're saying, Vincent. So now we're actually, getting some real headway into human population diversity. And, the good news here is not just in European populations, which have been certainly oversampled to date. And so, and now what we're looking at is, again, sort of thinking about time. So like what's been happening using these population genetics measures, what's been happening in the last 500 generations in terms of a selection coefficient. And this is, a measure of how much overall natural selection we think has been acting on this set of genes. And you can see that in both cases, that sort of clicks up a couple of points here, or hundredths of a point, with some error bars, but proposing a statistical difference
Nels,
again in that time range.
Could you do this with all the other genes? Because here they're using the VIPs, right?
Correct. Yeah.
And they're looking at that over generations. But don't you think they could have included this for all the others? Yeah.
I agree with you, Vincent. And so, I think obviously they're just sort of highlighting maybe the most striking finding, but I think that's kind of what I mean by taking a grain of salt with that definition of the VIPs. And so, what I would like to see actually is if you take any set of, let's say 40 or 50 genes and put them together and then run the same analysis. Like, do you start to see, is there just enough of, because we're not dealing in single genes here, we're doing kind of enrichments of a group. And so in some human population, just by chance, if you put together a bunch of genes, will there be a signal somewhere, in that? And so, that's where I kind of am, a little bit measured maybe in my enthusiasm at this point for this paper.
So what other kind of grouping could you do to check that out, right? So here we're these are VIPs, right? Should we use like VIPs for Ebola virus or something else just to see what happens?
Well, yeah. So there, I think, there you might be enriching, hopefully, and that's hopefully happening here, that you're enriching for sort of a specific set of proteins. The group has identified other VIPs, a bigger group, kind of not virus specific. And so you could even just take another set of those. There, that might, if you see a different pattern, you might say, oh, well, there's a different pandemic from a different set of viruses. So I'd actually like to see, maybe, so, and actually this is really helpful. So if we go ahead now to figure 6, to just ask, actually, how did this set of VIPs get identified in the first place? And so this actually brings in the mass spec. They were actually just confirming the interactions that were proposed for the VIP. So what we're looking at here, I won't drag you through the weeds. It gets a little bit technical, but it's so in the panel A, we're looking for the co-location of virus gene and host gene, and so the human proteins are tagged green. And then the virus proteins, SARS-2 proteins in this case, are tagged red. And if you see both bands in a gel, that suggests that there is a direct interaction between the host protein and the virus protein. And so if you actually scroll down just slightly, Vincent, we can see, actually panel C gives us a nice summary. So all of those blue balls in the middle there, those are virus genes. So we're looking at SARS-2 genes, ORF6, NSP2, ORF10, et cetera. And then kind of the spurs coming out are the proposed interactions with human genes. And so all of the green balls are cases where they could validate. So they're basically taking, this isn't in a cell, this is in a test tube, but they're taking the human protein tagged green and the virus protein tagged red and putting them together. The color coding now actually switches up on us in panel C. So it's actually the green balls are the ones they could validate. The red ones, they didn't see that sign of an interaction. And so already there's a little bit of sort of complexity here that not all of the proteins in the group, even some of them that are probably kind of enriched for these signatures of sweeps, may not in fact interact. Then you have to you kind of start peeling back the layers of the onion here. Even if these proteins, virus and host, interact in vitro, does this really play out in the course of a natural infection? We don't have, some validation and some overlap for sure. But that's another issue, is sort of the compromises of getting strong data for an interaction versus the complexity of a real virus infection. So anyway, so finally getting to your question, Vincent, I think, taking another set of, let's say, even not virus proteins, but just host proteins, and asking maybe, roughly the same. So what is this? Maybe 20 or so virus proteins, seeing host proteins interact, and then putting together sort of an equivalent list where you have no sort of enrichment for that you're proposing for viruses. Is there selection that happens there at a level that meets this bar in different ways? If the answer is yes, then I think we have to be a little bit cautious about overinterpreting these signals or overfitting that what's really happening here is all virus related or pandemic related, and are there other forces here? Things involved in reproduction, which is also another major source, obviously, of natural selection. If you have a set of mutations that for some reason mean that you're reproducing more, regardless, and that could involve microbes somehow and virus infections, but outside of that, right, that's I think a little bit of an open question.
So, I mean, it's a striking peak, right?
Yeah, agreed.
And did they use any other the genome sequences besides these East Asian ones?
They did. So they did a whole different, just as you're saying, this really fun opportunity to do European populations, to do other populations, and that's one of the findings that they hold up as being novel, is that it does really hone in on East Asian populations. Now it's important here, and actually the authors themselves in the discussion offer some caveats as well. So they note that the results do not represent evidence for any difference for either increased or decreased genetic susceptibility to coronaviruses or to SARS-2 in particular in any human population. And so they're a little bit careful, to measure this. You're looking at you're trying to speculate, or not speculate, you're trying to infer sweeps in certain populations, but it doesn't have that resolution. So I think it's, tentative enough that you can't say, okay, because of these sets of sweeps, that means that this, population of humans might be more or less vulnerable to a virus of today because of what they faced in the past. So that, that probably is true. Certainly theory would kind of move in that direction, but we still are kind of in our infancy of putting together the specific changes that we see in populations and how that leaves you vulnerable or protected to the kind of pandemics of today or the pandemics of tomorrow.
But Nels, these viruses don't just remain in Asia, right? They sweep globally. Like the 4 common cold coronas are everywhere. So why didn't we see this sweep in other humans besides Asian, right?
I love this question. So I think what you're saying is exactly right for modern human populations. But I think it was if we go back 20,000 years ago, there before the advent of the 747. But more seriously, right, how much, so human migration really matters here, and human isolation. How big is the total population, and how much sort of geographical mingling is there? And so, so that could even run for generations where you might not see, a population might not interact. We've scrambled the deck completely these days with globalization. But I think that's still, that you would predict in our
true.
deeper history.
So 15, 20,000 years ago is when people started to congregate in bigger and bigger cities. So maybe this is just about the right time where you have some large gatherings of people, yet it still doesn't get to another continent or something. So a good control would be to do this for a virus that emerged later and spread globally, right?
Yeah, I like that idea. Here's a couple of tricks, though, that immediately are obstacles that immediately come to mind, right? Is that viruses themselves, even different classes, I would say, don't necessarily, they're definitely episodic, but they're always kind of hanging with us, right? And so the authors do they aren't trying to say, there was SARS 0.1 or something 20,000 years ago, but they do kind of say, well, maybe there was a coronavirus on the basis that these genes are, interacting. I think, if you took VIPs, though, with other major virus classes, you'd see a pretty big intersection of this set of genes. So that would be, I think, an interesting, we already kind of hinted at that, right? Is to sort of scramble the deck. Now, that's a lot of analysis, so I'm not trying to, overburden for one study. I mean, there was such clean peaks, or such a clean peak in that figure 2 that we looked at. I think was sort of the biggest surprise here, that you could actually, that there could be some signal lurking there at all. But the viruses, the idea, certainly from theory, is that, a pandemic happens and then there's another one kind of in the offing. And so you're never there's not like long terms of kind of quiet between pandemics that they're ongoing.
Nels, when would we expect to see such a sweep caused by SARS-CoV-2? How long would that take?
Yeah, so I think we're really, that's one of the great questions about some evolutionary context here. So, for example, kind of in this traditional sense, our species, SARS-2 has been awful for many of the reasons we've been stepping through for the last year and a half. But our species, we're in no danger of kind of extinction level events where you could imagine that entire populations are being wiped out. It also, I think it really depends, on the virus, so mortality by age for the virus, right? And so a virus that has more mortality in post-reproductive age hosts versus pre-reproductive age hosts, that will have a very different sort of impact in terms of a sweep. There has been, and I don't want to pull that off the table altogether. So for some populations, the HIV pandemic has been proposed to be, there's enough in some localities. There are, certainly not entire species extinction level events, but enough, tragically, enough, entire villages being wiped out in a sense. And so there you could imagine that there's something like a soft sweep, a partial sweep or a pretty, drastic impact on who survives, and has led to some really interesting ideas about the sort of elite suppressors, just someone who has a genetic predisposition where they're not is vulnerable to HIV. So, famously that deletion in CCR5, the HIV receptor, so that the virus doesn't see the cells that it needs to infect. And then we're back to the arms races. So the viruses then evolve the ability to enter cells without needing CCR5. And there are certainly strains of HIV that have those properties, and you're kind of evolving back and forth again.
Of course, the Delta CCR5 was present before HIV emerged. So what led to that sweep, the sweep of that deletion?
That's right. Well, and that's kind of that's the hope of this paper, right? Is that it's sort of teasing out that ancient signal that happened 20,000 years ago. And maybe it was in that one that you selected. And so that gets to the sort of ultimate goal that you would love. And they are kind of, stepping up to the limits here. But does this result in genetic differences for either increased or decreased susceptibility? And that's maybe for the work ahead.
The other aspect of this paper, which we could just cover really quickly, is people have done GWAS analysis for SARS-CoV-2 to see what SNPs or which polymorphisms might give rise to more severe disease. But unfortunately, they haven't been done in this population, right?
Yeah, or that work. It's sort of like the GWAS. We're not doing sort of that deeper dive into evolution. We're not, you're not recreating those recombination graphs, sort of the working backwards in time. You're sort of asking what's here today. So there have been, some. I think there's a couple of papers on this antiviral protein that we have called OAS1, which is short for oligoadenylate synthase 1. And there's some interesting splice variants in human populations. And then, as people have linked this in some cases to the ancient genome sequencing done in some of the Neanderthal samples, and you can actually see some of these splice variants in some of our ancestors even before humans. And so the idea is that, our genomes were whatever, on average 2% Neanderthal or something, have some of that genetic variation that arose in those populations and then got introgressed as our species interacted and ancestral species interacted. And then this today actually echoes on, and people say, we can thank the Neanderthals for some of the variants of OAS1 that might provide some protection against severe. So you'll still get infected by SARS-2, but you're less likely to have a severe case. And so just glimpsing at some of the first ties along those lines that you're describing of GWAS to kind of working backwards in evolution.
That was a paper we did on TWiEVO, I think, some time ago, the Neanderthal OAS introgression, right?
That's right. And then the kind of fun recent echo on this, not literal echo, but scientific echo.
No echo.
No echoes. We're done with echoes today. Is that there's in OAS1, this one of these, I think a different variant, where you have a lipid or a prenyl, basically part of the protein that will attach to a prenyl group, a lipid group that inserts the protein into the membrane and gives it some localization and seems to kind of put it in the right place at the right time to give you sort of a better defense against SARS-2 as it's replicating. And so, there's kind of these interesting connections that are starting to just bubble up a little bit. All right. So that covers our first letter. So thanks again, Mark, for recommending that paper. This is one that's been on my radar for a while. And I think it kind of raises some provocative points and also, maybe a note of caution that there's a lot of kind of new techniques and sort of categorization of genes here that we're thinking about the implications of that and how well some of these patterns hold up with some more scrutiny in the future. Okay, so then Vincent, our second letter in the TWiEVO mailbag, this is from Daniel, and he writes, "Hi TWiEVO, I'm a big fan. Although I hope that you can soon share some more non-COVID related research. Cheers and thank you." And so Daniel included a link to a pretty interesting paper. This is kind of goes a little outside of our wheelhouse, I'd say, Vincent. This is thinking about how evolution might be acting sort of not just on a true, like a kind of, solid biological feature like the evolution of our immune systems, but how evolution might be acting on human culture even. And so how do we how do more complex behavioral outcomes maybe sort of through the lens of evolution? And this is, I think, a fascinating topic, one that we're punting on, so we're not going to cover this topic today, although appreciate maybe in the future we can bring in some more experts on these complex evolutionary questions. We're going to go in a different direction here, but a non-COVID direction at Daniel's suggestion here. And so we found a preprint that caught both of our eyes, and the title of it just dropped maybe a couple weeks ago, is Membrane Permeability Differentiation at the Lipid Divide. And so
I love it, Lipid Divide.
Yeah, isn't that a great title? So that's, so maybe even a little bit mysterious, like what is a lipid divide? What are we is there even evolution going on here? And so let's stay tuned for that. We'll unpack that in a moment, but I just wanted to lift up the authors here. And so again, an international group. This is mainly coming from the University of Oxford in the UK, with collaboration with some folks at UC Santa Barbara as well. And so the group here, which is in the Living Systems Institute and Biosciences at the University of Exeter, this is first author Ursula Lipinska, And then senior co-authors, co-senior authors, I guess you would say, Thomas Richards and Stefano Pagliara. Thank you, Vince. I was just waiting.
Soft G.
Appreciate it. Thanks for the Italian interpretation there. And so, so let's go to the lipid divide. This is important. So there's this idea that really interesting idea, important idea, that in the deep branch of life, the split in the tree of life between what went on to become modern bacteria and what went on to become modern archaea, this split also could be sort of defined by a lipid divide. And so this is actually thinking about the membranes, the packaging, the compartments for bacterial and archaeal genomes. And it turns out that there's like very different phospholipid bilayer compositions in all of archaea relative to all of bacteria. And so this is important because around this time in early evolution, it wasn't, relatively speaking, it wasn't too far before actually sort of the last common ancestor, the Luca, the emergence of cellular life, and then with another split, of course, to the eukaryotes, which is the lineage where we find ourselves.
Here we go.
So here's the Wikipedia version of tree. I'm trying to remember if this has been updated. It probably has a couple. There are these, great debates, these controversies. Are there 2 branches? Are there 3 branches? So here we see the Great Divide happening where, purple and red go apart, and so the exact relationship with the eukaryotes is probably still being litigated scientifically in some circles. But this does get to that point, right? Which is you don't have to go too far, where we see the cursor that Vincent's pointing out, and
this is Luca down here, right?
That's that's the idea. Exactly.
That was an Italian cell, Luca, right?
That's right. Emerging from the primordial soup here somehow. Okay, so to kind of get back to the evolutionary story, then, so as these compartments are coming online and you're taking some set of nucleic acids, and there's fascinating biology here. So the idea that the initial nucleic acids was RNA, that DNA potentially came much later. So the RNA early hypothesis, and there's some evidence supporting that, and that RNA can both encode, you can code for something, those 4 letters of the genetic code, but also you can fold up RNA and it can act enzymatically itself. And so it's sort of, before you segregate your coding, sort of the blueprints from the actual construction of the proteins that make up the cell or part of the cell. So the big question here, kind of related to this, is, if you put up a barrier, a cell membrane, how do you get the metabolites across, right? So you have to, if you have 2 closed off of a barrier, you're not going to get, as a single cell, you're not going to get the raw materials you need to propagate for life. So things like amino acids or nucleic acids in particular in those early days, if we're talking about RNA. And so so that was the question here, is actually, if you think about the lipid divide, is there a difference in permeability, or a comparison point to be made that might start to hint at sort of these transitions in permeability? And, if we fast forward all the way to today, actually we know, like for our own cells, the permeability is quite low, but what do we have? We encode all these transporters. And so we put proteins into our membranes, into our lipid bilayers that traverse that and then offer sort of a traffic route for metabolites to come in. We actively transport, or in some cases passively transport, these things across ion gradients, for example. So a lot of complicated biology that came on, but we're kind of trying to rewind the tape in a sense here, or the researchers are. And so they actually claim that so the lipid divide has been known for a long time, and it's actually the conventional wisdom was that bacteria and their membranes were much more permeable than those of archaea. But they lift up that no one has really kind of tested that in real life somehow. And so here's where I think some clever technology. So they actually built some microfluidic devices so that they could actually, set up membranes, set up vesicles, little compartments that were either composed of lipids that were archaea-like, or were more bacterial-like. And so there's a caveat or two there in that these aren't actually bona fide membranes from these living critters, but do have some of the key fundamental properties that are different between them. And then, there's a great, I don't know if you can pull this up, Vincent, if you have it, the first figure, which shows the device that they use. I mean, this is just really kind of a picture is worth 1,000 words here for sure.
Let me get it up. Yep.
Yeah. And so the whole system to begin with is, here, perfect. So if we look at panel A, right, so there's this inlet for wash buffer, and then there's a metabolite inlet, and then an outlet. So the solution is flowing from left to right.
Yeah.
And then you can see these little dots. These are the vesicles. So these are as they're building them kind of in situ in the microfluidic device. And that green dye gets incorporated when they actually have a viable vesicle, a little circle or sphere of membranes. It traps that green dye, and then they just push a washing buffer through there. And so in panel B, we can see before the experiment is running, one of these little dots, it's the white dot in the middle there, that's actually a fluorescent dye that remains as the external dye is washed away. And so then they do the experiment. So they take, in panel C, from that bottom inlet, they start taking, again, a different fluorescent label, but they begin to move metabolites across the membrane here. And they picked, as we were kind of just mentioning, they picked different ones. So some of the amino acids, some of the nucleic acids, the building blocks, uracil, for example, one of the 4 nucleic acid bases, the U that you find in RNA. And then the test here is whether or not that fluorescently, as indicated in blue, whether these fluorescently labeled metabolites go inside the vesicle, and then when they wash that clean, you would have a signal that would remain if it's permeable, if it's in there, or it would be excluded if it's not. And so then they just march through, and they do this for the bacterial, half a dozen or a dozen metabolites on the bacterial vesicles, and then the same set on the archaeal ones. And the surprise here that they found is that they actually saw more permeability in the archaea than the bacteria. So this actually is a 180 on the lipid divide. It sort of flips the conventional wisdom. And you see that across the board, actually. I think kind of the patterns that show up for all of the metabolites that they test. So they do a little bit more work. They actually kind of go back and try to nail down some of is the permeability they propose is because of some of those linkages that the lipids have. They get methylated, or just slight chemical modifications that they propose or have been proposed that give archaea, in this case, this property of being a bit more permeable. So then it's kind of a simple paper here, but the last step is a cool kind of correlation. So, and I kind of previewed this. So mentioning that we encode all of these transporters, metabolite transporters, in order to get through a relatively impermeable barrier. And so they kind of march through the ion transporters, the amino acid transporters, the nitrogenous base transporters, sugar transporters, and then, again, a phylogenetic collection from a ton of different archaeal genomes and bacterial genomes. And what they show is just if you just do numbers, like if you just count the numbers relative to total proteins, you see fewer transporters. The genomes of archaea are less dedicated to encoding transporters, whereas bacteria encode more. And so that kind of fits the notion that, well, you don't, if you're already permeable, you don't need to transport things across. It can kind of make it on its own. And so I think it's kind of a simple result, a simple correlation, but I think it does raise some kind of really provocative questions. It got me thinking about what's going on here. So when you think about, and remember, it's not that the archaea are more quote unquote ancient. There is a single split kind of near the trunk of the tree of life. But then both the modern bacteria that we see today, the modern archaea that we see today, the modern eukaryotes like you and me that we see today, have all had the same amount of time to evolve. And so it's not the notion of a kind of a primitive or a quote unquote modern state, to be a little bit careful about thinking about that, but thinking about how could these systems maybe take different routes? And so that got me thinking kind of on two ideas, Vincent. So the first is that it takes energy, right, to encode transporters. And so if you can tolerate some permeability, maybe there's an advantage there. You don't have to sort of have this extra complexity where a good portion of your genome is dedicated and the energy is put into the transcription, translation, the building and the trafficking, et cetera, all of this stuff to get the transporters in place to get your metabolites in. So that's one idea that kind of hanging loosely. The second one I wanted to put out there is, what happens when you put transporters potentially on the surface of your cell and it's a target. So it can't help but circle back to viruses. What do viruses recognize when they, in many cases, it's actually the proteins that we put on the surface of our cells that then become receptors. If we kind of harken back to our last paper, the VIPs, these are receptors or transporters in some cases, can be virus interacting proteins. And so that could be an interesting difference in the history of how viruses even were sort of interacting, I think, with cells, or whether they could gain access to the nucleus to mount productive infections. And so if that's true, could there be some clues here, not just about sort of the composition of ancient membranes, but even what might have sort of set these sort of major branches in the tree of life in different directions? Is there even selective pressure to avoid transporters because you have fewer targets for virus entry? So anyway, this is a totally half-baked hypothesis, but it got me thinking. So I did a quick search, just, what's known about archaeal viruses. And we're kind of archaeal viruses, the viruses that infect archaea, and kind of at its infancy compared to most of virology.
It's true.
For sure.
It's true.
But some really interesting, early sightings here. And so I found, just a quick search, found a great kind of review or preview piece in PLOS Pathogens. This is the Pearls section. And this is actually edited by Kathy Spindler, one of the stars, one of your co-hosts on This Week in Virology. And so Kathy edited a piece by Jennifer Worth and Mark Young. They're at Montana. And the title is The Intriguing World of Archaeal Viruses. And so we'll put up the link on the website. It's a really short piece, but it just sort of highlights. So there's certainly some viruses of archaea that have some of the same kind of characteristics as some of the virus groups that we're more familiar with, the bacteriophage and bacteria, and even some of the eukaryotic viruses. But there's also a lot of different and seemingly, weird biology going here. And I wonder how much of this might actually be connected in some way or another, whether it's a direct link or not, who knows, but to the differences in membranes. The differences, the first thing that a virus sees when it might infect a new cell, when it might infect a new host. Could there be some clues there at that lipid divide about how viruses behave differently?
Well, one of the reasons that we don't have a lot of virology work in archaea is that they're extremophiles, and a lot of them grow at high temperatures, and it's hard to do. Do you know Ken Stedman?
I do.
Over at Portland State, I think is the name.
That's right.
He was on TWiEVO a couple of times to talk about this. It's really interesting. You have to have water baths with polyethylene glycol in them because they go above boiling temperature, right? And water would just evaporate and so forth. But it's an interest. It's totally understudied and needs more for sure. But Nels, this idea, I don't understand. Then the bacteria membranes are less permeable, right? So
correct. Yep.
Perhaps at a different time they were more permeable, right? Earlier, and today is not a good reflection of that. So the different composition between bacteria and archaea is today, and we have no idea what it was like billions of years ago. And so this doesn't really negate any evolutionary ideas about prokaryotes and archaea and so forth, right?
Oh, not at all. I think the simplest hypothesis would be that, in as you lose permeability and have to bring in more of this biological complexity, the transporters that are encoded in a DNA genome, you wouldn't have those at the beginning, right? And so the idea would be that even though the archaea are modern today, they're a little closer in character. Their membranes are a little closer in character to what might have been sort of the original cells before that, before the lipid divide, before the first major branch in the tree of life. And so, no, that's the idea here. And that, the permeability, or the kind of the echoes of the permeability in a sense in archaea might give some clues about how, because the, sort of attractive feature here is that you can bring in lipids, but because there's enough permeability for things like nucleic acids, that you can have sort of the building blocks before you get to this more complex scenario that you see in the bacteria, where you need to bring these things across with transporters in more cases, as proposed by that correlation. Yeah.
I think it's a really interesting approach, because you use good technology to address fundamental evolutionary questions, right? You don't just think about it and hypothesize. You actually do experiments.
No, I know. And that's sort of like a double surprise, right? Is actually that, there's the idea of that divide, but no one had really that was, I think, noted by some just early measurements of like, here's a sampling of modern species, but then to actually, as you're saying, to do the experiment, to set up, use the microfluidics, set up the thing, and then actually do a test for permeability in real time. And so, and this is like the reason I really thought it's fun to kick this around a little bit, is that, it's really tough we're talking about, things that happened 3 billion, 4 billion years ago, something like this, right? And so how do you even design an experiment that really kind of gets at that? And there's, I think, all kinds of work nibbling at the edges in different ways. And so anytime someone kind of comes up with a clever scheme, not to like definitively like recreate or settle the thing, but it just sort of, again, sort of provokes your thinking in a new way when you put something into action, when you gather some evidence, even if it's sort of peripheral to that. And so that kind of harkens back to, what brings you and me to the table here on TWiEVO, right? Is we're trying to find these sort of attacks, or these like, these experiments that sort of take these seemingly intractable questions and sort of put it in a new framework by just trying something and seeing what you get.
We should some point do a paper on, this idea that there was a precellular stage where there were self-replicating nucleic acid-like molecules, and people actually do that chemistry, and they can get it to work. You can get short, RNA-like molecules to reproduce in the absence of a polymerase. We should find one of those and do that.
Absolutely,
yeah. Or maybe get a guest who does that. That would be
cool. No, that's right. Or a guest that might write about it. So part of talking to Carl Zimmer earlier this summer in his book on, what is life? How do we define life? It hits on, some of the chapters hit on exactly these kind of experiments, and even put together this really cool timeline of how science, for the last roughly 100 years has kind of tackled this question in different ways. And you actually start to see where you go from kind of the arguments into some experiments, and then exactly the kind of work that you're describing. And then even these spin-offs where, you're asking these fundamental, almost seemingly intractable questions about what was happening in a mud puddle 4 billion years ago. But then by thinking about permeability and transporters and channels, all of a sudden you think, well, wait, maybe we could even use that. And so that's one of the fun kind of parts of Zimmer's book on life, is how actually some of our long read sequencing technologies borrowed from some of the insights about thinking about these questions. And so it kind of takes on a new life of its own and facilitates science moving forward, unexpected advances.
So Pete asks, so archaea aren't older than bacteria despite the name?
That's right. We would put them, as we're working here deeply in the branch, it all starts with sort of one population, and then with this big divide. But that means that the age is the same, even if the features might seem more. And that kind of gets at this great, paradox sometimes. Like, so in the way we talk about sort of primitive things relative to modern things. And so the notion of a living fossil, which is kind of a dangerous construction. So people talk about the coelacanth fish as a living fossil, but it's evolved just as long as modern. It's the same age, evolutionarily speaking, as every other modern fish, every other critter out there. And so it might have some characteristics that we associate with the fossil record, with things that existed a while ago, but evolution has happened just as much.
Yeah, yeah. Krister asked, are there any emulsifiers in these archaic membrane soups?
Oh, that's a great question. Yeah. So, and I'm going to have to take a pass on the this kind of goes beyond my working knowledge of. So, what is it? And they do address this to some degree. So, what is it that gives these phospholipids a more permeable character? And so, it's some of the modifications, the methyl groups, I think, in the tails of the lipids and the carbon chains that might be part of it. Yeah, whether there's other emulsifying agents in there, I think it's a great question. And might even, so the nice thing about these preprints, right, on the bioRxiv is this is, dig in yourself. And, if you see something along those lines, let us know. We're here learning in real time the same way you are. And so that's the whole idea here of this conversation.
He also wants to know if the lipid membranes repolarize, thereby removing membrane proteins.
Oh, interesting. Yeah. So that's a whole nother interesting field here, right? Is and since these are bilayers, you also have this more complex activity. These things aren't just sitting in a static. So first of all, the polar head groups are moving around like crazy. You've got all these kind of tails entangled, more or less entangled, depending on how saturated, how you've modified it in these cases. And then there's also flipping going on here. And in fact, there are enzymes that that show up and associate with the membranes, and including in eukaryotic membranes as well. And so there are some great ways of actually doing labeling, both on the so they're just labeled, or didn't even label the lipids as far as I know. They just have a fluorescent dye that to see that they have an intact sphere, an intact vesicle. But you could take the system to the next level, which would be to either directly fluorescently label either the interior polar head groups or the exterior head groups. And then now you can start to get even a more resolution on those dynamics. And pretty soon these things are flipping around. And in most cases, I don't know for the archaea specifically, but you end up actually moving, the fluorescence goes to the inside and the outside and back, and you end up sort of homogenizing that population.
Mr. Ozzycam sends us a link to a paper which is called The Essence of Life Revisited, How Theories Can Shed Light on It. And this is from, what's the journal? I don't even know. Theory in Biosciences.
Okay, yep. This is a new article.
Yeah, disagreement over whether life is inevitable when the conditions can support life remains unresolved.
Yeah, I love it. Yeah.
So this is kind of relevant to what we're talking about today.
Yeah, agreed.
For sure.
Yeah, definitely. And this gets back. So yeah, like that's, I mean, I kind of spend my professional life on the tips of the branches of the tree of life, what happened in sort of evolutionary as blink of an eye. And so, yeah, this idea of, if as non-living systems self-organize, is life inevitable? What an incredibly exciting, great question, and one that just also scares me. Like, how do I tackle that? That's why I really like this preprint, because it's like, kind of makes just a little inroad. Doesn't like recapitulate that branch of life, but it sort of makes a little inroad and just kind of provokes some thinking in that space that I think actually can cause some progress. And so I bet there are some similar ideas put forward there in that review. Thank you for sharing. I'll add that to my reading list as well.
So we had a colleague of yours on Immune, Nels, Kiki Fairfax.
Oh, fantastic.
Do you know Kiki?
I know her well. I sit on the thesis committee of one of the students in her group, and we've been circling around thinking about how can we collaborate in more depth.
She's terrific. Yeah. She's fabulous, man. Just terrific interview, and her work is awesome. She really, I loved it. It was really good.
Oh, fantastic. I'm glad to hear that. I'm a big fan of her. She's one of my science heroes here for sure. And so happy that she's a colleague here at Utah, bringing all kinds of life and energy to the immunology that she does, and looking at these, these parasitic worms and how, like, whether the mother is infected, what will that mean for the offspring? and starting to look at this kind of
It's very
pulling
cool.
this apart in really deep detail, epigenetic detail. And
Makes a big difference. And we now know that we did a paper on TWiEVO last week that if the mother has SARS-CoV-2, the immune system of the baby is affected. And
interesting. Yep.
We don't know how. I mean, is it cytokines? Is it other molecules moving across it? Kiki thinks egg proteins from the schistosomes are going across the placenta. So very cool stuff.
And I agree, sets the table. This is, again, these sort of confluence of tools, and to get at like a more complex view of biology. This is sort of the fun that people are having right now with these great questions.
Nels, you want to do some picks?
Yeah, let's hit our science picks. So I'll kick it off here. So I just found this link, kind of came across my table. This is a paper in PNAS, and I think basically the take-home message here is actually don't publish too many papers. This is an ode. I'm claiming this as an ode to slow science. So let me set this up here. We'll, of course, include the link in our show notes. But the title of the paper is Slowed Canonical Progress in Large Fields of Science. And so basically this is a survey. I think it's across many subjects, not just virology or evolution, a lot of academic fields. And the idea is that, if you look at the millions of papers out there, this deluge of information, that more isn't necessarily better in terms of scientific progress. So this idea of kind of quantity, if we just put a lot of information out, that somehow science will march forward, will make progress, we'll have these insights into how life around us works, or how, various academic fields make progress. And I kind of, so I have to confess, I haven't looked too deeply at like how rigorous the data set is here, or what even the specific measurements are. But I really like some of the ideas, at least intuitively. I think they really resonate with me. So I think, as in many fields, we actually do this kind of counterintuitive thing, which is that more is better. And in fact, careers can rise and fall on this idea that, have you published enough? Have you put enough out there? Publish or perish is sort of one of the cliches that we hear in academic science. And so, but I think this idea is right, that you can just sort of flood the zone with information, but are you adding value? And if things get too competitive, if you have a great new idea that might actually, if someone saw that, it might break through and advance understanding, advanced thinking, or sort of cause humans to ponder something in a different way. It just kind of gets lost in the noise. And then this leads to, what the authors are talking about of, this sort of, you're kind of stuck on these old ideas, and you're just sort of adding to the edges of it because you're just trying to get stuff out there. And the things, you ossify, the whole thing becomes calcified, and things don't move forward. So anyway, that's my science pick of the week.
I like this.
Slow science.
It leads to, the more papers leads to ossification of canon.
That's right.
Do they offer any solutions? Because you can't stop people from doing work and publishing, right?
No, you can't. But I think what you could do is actually challenge some of the sort of conventional wisdom that more, and to say actually maybe more isn't better. And we do this both in terms of quantity, so how many papers has. So when let's say a professor at a research institution is on the tenure track and coming up for tenure, or any kind of job promotion, really. And if part of the assessment process is a literal counting of the number of papers, it's also, but it's not like a simple solution of just saying, and I think the authors mentioned this. Well, you could, instead of saying the number of papers, let's talk about quote unquote high impact papers. But that offers a whole nother kind of layer of complexity, or of complexity, because what does high impact mean? And that definition can get bent in various ways. And so I don't know, man. I think I think science shows, podcasts, or online courses, this is a way of breaking through in a different way, right? Is where people are saying, hold on, here's, in a flood of information, here's, not the definitive word, but here's a couple things that are worth keeping an eye on. Here's something that's interesting. And so, in addition to this, I think we are seeing maybe some hints of this in peer review, where basically scientists, certainly in our kind of fields around where we work, are starting to try to reclaim what it means, the peer review process even. So you put your work out on as a preprint, and then, it might get picked up on a podcast or a science show, or someone might, use that for a journal club or something. And so I think it's kind of goes back to some of these more kind of word of mouth and what are the things that just really capture people's imagination and sort of pushes the conversation forward. Now, I don't know how to formalize that. Like, how do you value that? You can't.
Yeah, that's the problem.
Yeah, that's the problem. And that's a real challenge.
Well, we've gotten ourselves into this situation because, as you say, promotions, grants, speaking engagements, it all depends on publishing. And the more you publish, the perception is you're better. But, it's not true. But I think it's very hard to reverse that kind of process,
Absolutely. Yeah, that's right. And so,
you publish one paper a year, Nels, if it's really groundbreaking. They'll say on your next NIH grant, they were not productive.
Yeah.
Can't win.
The definition of productivity, if we can somehow break through. I mean, so as peer reviewers ourselves, if we can point out, like, wait, hold on a minute. Let's say you're reviewing a grant and someone, launches that as a criticism to try to stop the conversation from it and say, well, hold on, what do we mean by productivity when this paper, maybe it's one paper, but it's changed the whole trajectory of a field, or it's taken that ossified cannon and flipped it 180 degrees because of a new finding. And so those are sort of the cracks, hopefully, that we can advance. So anyway, that's my pick of the week. But Vincent, I'm excited to hear about your pick of the week because I was going to ask you about this. This is I'll let you say it.
My pick is my Virology Live, which is a live streamed virology course. Now, about almost a year ago, Amy and I started live streaming a Q&A, and then you and I started it. And I really like it because of the people in the chat. They make it interesting, right? They're here. They want to hear scientists talking. They have great questions. They interact amongst each other. So I, at some point, thought, why don't I teach a course this way? And so I started a couple of weeks ago. I say, I'll call it Virology Live. It's based on my Columbia University course. It's 2 hours, Monday and Wednesday. And we're going to have 25 lectures of all the basic virology, as I usually do. But it's the chat that really is cool. People are chatting amongst themselves, which you can't do in a class, right? You got to be quiet in a class.
That's right.
I pause periodically to give them a little quiz, and then I answer their questions. So what normally would be an hour and 15 minute lecture is 2 hours, basically, because I'm answering questions. And people seem to like it. In fact, a lot of the people here today were at this morning's session. So I'll give a link in the show notes and check it out. It's over on YouTube, 11 a.m. Eastern, Monday and Wednesday. And it's recorded, of course, and it's on YouTube, so you can always go back later and watch it and also see all the questions and so forth. And I provide the slides as a PDF. I provide study questions. There are weekly quizzes. And there's going to be a midterm and a final. So
I love it. It's so good.
It's an experiment.
Hey, no, it's a great experiment. It sounds like it's off to a great start. I'm wondering, one question I had was, are there, do you have scientists showing up listening in, or maybe making cameos? Are scientists invited? Could I show up and join the conversation in the chat, or how are you?
You could certainly join. There are probably some scientists showing up whom I don't know, or who, they're there. You could certainly go, but you might even want to give a chat, give a lecture, give a session. I call them sessions. I try not to call them lectures because I call them sessions, and it's more of an interactive thing than a classroom can be because you can really go back and forth. So you can check it out anytime.
I think I might take you up on that. I like this idea of kind of improvising in real time. Certainly, it can go in the wrong direction, as we worked out our echo at the top of the hour here of our podcast, but can also really bring some interesting new conversations to.
It was an experiment, Nels, and I said at the onset, if it works, I will do other courses that I don't teach at Columbia. So I would do sometime next year, a course on viruses. It would be Viruses Live, and I would go through the major viruses and talk about them, which is not what we do in this one. This is a principle-based course. Gotcha. And I would do other things like emerging viruses, right? Go through what we know about all the emerging viruses throughout time. I mean, so many things I could do. And that's also why the incubator is cool, because I can do it all here and have a lot of flexibility. So I'm having fun.
Wow. That sounds like a bunch of fun. Virology Live, Mondays, Wednesdays, that's at 11 Eastern?
That's right. That's right.
Very cool.
And
Very cool.
Now, the cool thing is getting people from all over the world. I mean,
that's the best.
Some people have to get up at 1, 2, 3 a.m. to do this. And I have lovely moderators, many of whom are here today. Mr. Ozzy Cam, and he's in Australia. I think it's 1:30 a.m. for him when it starts. And also Steph SF is a moderator. And some other people. I think Les is here. Yes, he's also a moderator. And so look at that. They were this morning, and now they're here again. How cool is that?
Really cool. I'm glad we had some virology to kick around here, actually. We're staying on topic. It's hard to avoid the viruses. They hold the keys to a lot of evolution. They hold the keys to a lot of the life around us.
So thank you, Sandy, for your contribution. Really appreciate it. And I hope you see that we can solve problems in real time, right? I had this issue. And fortunately, Nels took it away, and I wandered off and sorted it out. And this morning we had an issue. I sorted it out. So I'm not saying I'm great, but I can fix it. I'm pretty sure I can fix it. So thank you for your support there.
Yeah, thanks a lot.
Yes, the teaching works, and here's the link Steph put up to the course. There's the syllabus, the teaching works. So I'll teach more courses. You got it. As long as I'm around, I'll be teaching courses. And at some point, we'll have Nels come in and give a guest lecture. That would be fun, right?
Count me in. Guest session.
Guest session. Sorry. Guest session.
Yep, kind of come in and improvise a little bit. We can play some science together. Yep.
All right. That'll do it for this live stream TWiEVO number 72. You can find the show notes at microbe.tv/TWiEVO. If you want to send in a question or comment, it's TWiEVO@microbe.tv. And if you really like what we do, now many of you have supported us today in a super chat. We really appreciate that. There are other ways you can support us. You can find that out at microbe.tv/contribute. Nels Eldies at Cellvolution.org. There you go, Cellvolution.org. And he is on Twitter, L Earlybird. Nels, good to see you again.
Hey, Vincent, good to see you. Glad we ironed out some of those wrinkles as we echoed through our podcast today.
Yeah, it's the first live stream I've done with a guest, and so that is a different situation from me teaching.
Absolutely.
And so this is the same exact thing that happened when I started the Q&A with Amy almost a year ago. I had to echo. It was at home. I had a different mixer. I fixed it, so I have to work on that one. I'm Vincent Racaniello. You can find me at virology.blog. Music on TWiEVO is by Trampled by Turtles. You can find them at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious. Thanks, everybody. Bye-bye.
Thanks, everyone.